Hello, everyone. Thank you for joining us today for our Molecular Connectivity Online Series, or MCOS. I'm Xin Di from New Jersey Institute of Technology. In case you are new here, the Molecular Connectivity Working Group is a group of researchers that aim to advance molecular imaging as a tool to studying brain connectivity. We organize this online series as a platform to exchange ideas and promote research on molecular connectivity. You can follow us on Twitter or LinkedIn and go to our website and subscribe to our newsletter. Today, I'm very happy to introduce our speaker, Dr. Jingyuan Chen. Dr. Chen is an assistant professor at the Martineau Center for Biomedical Imaging at Massachusetts General Hospital and Harvard Medical School. Her lab advances multimodal dynamic functional imaging techniques with an emphasis on ultra-high field fMRI and the functional PET-MRI, and the computational approaches to investigate the biophysical and molecular mechanisms underlying intrinsic brain activity, as well as the neurobiological consequences of cognition, arousal, and diseases. In today's talk, she will talk about simultaneous EEG-PET-MRI identifies temporally coupled, spatially structured hemodynamic and metabolic dynamics across wakefulness and NREM sleep. I would also like to introduce Dr. Claudia Brambilla. She is a new member to our group. She is a medical physicist and a research scientist at the Research Center Jülich in Germany. Her work focuses on nuclear medicine, imaging classifications, and clinical applications with PET-MRI. Once again, thank you for being with us, and let's welcome Dr. Chen's talk.
Okay, thanks, Ian. Let me share my screen. Okay, can people see my screen okay?
Good.
Okay, cool. So yeah, thank you for inviting me to present my research at the Molecular Connectivity Online Series. I have been a follower of the working group, so it's great to have an opportunity to present my research here. And I will jump right in. So today I will share our recent work that applies simultaneous EEG-PET-MRI to track different aspects of brain physiological dynamics across wakefulness and non-REM sleep. Okay, so it is well established in the animal literature that sleep is accompanied by substantial changes in various metabolic processes, including oxygen metabolism, glucose consumption. And in humans, brain dynamics during sleep have also been extensively studied using simultaneous EEG-fMRI, and a common observation is that during non-REM sleep, particularly in the light and intermediate phases of sleep, the amplitude of fMRI-based hemodynamic fluctuations increase markedly. And these large fluctuations also become more spatially coherent. So there we can see apparent increase in the functional connectivity. So despite a large body of work mapping sleep-related metabolic and hemodynamic changes, these physiological processes are often assessed in different imaging sessions. So this will pose some challenges for evaluating their spatial-temporal and dynamic coupling in real time. So the goal of this study is to characterize the spatial-temporal relationship between brain functional and metabolic dynamics across different arousal states. So specifically, temporally, our neuronal hemodynamic and metabolic processes were coordinated during the descent into non-REM sleep. And also spatially, what are the brain-wide metabolic changes support the altered patterns of functional activity during sleep. So to address these questions, we basically measure everything within a single scan. So with EEG, we can obtain ground truth delineations of each subject's arousal state. And with fMRI, we can map whole brain hemodynamic fluctuations, which indirectly reflect functional activity. And finally, to capture dynamic changes in glucose metabolism, we use the novel functional PET-FDG method. So I believe for this audience, you have heard about fPET-FDG from Christina Herford and Hamish Deary's talks before, but I here just give a quick recap. So briefly, fPET-FDG is a technique introduced by Jacob Hooker's group at MGH about a decade ago. And we know that in conventional functional PET studies, we have a bolus injection of FDG tracer, and then we compare the cerebral metabolic rate of glucose estimated from two separate sessions, one task scan and one baseline scan to infer task activation. But for fPET-FDG, it replaces the bolus injection with constant infusion, or sometimes with bolus plus constant infusion, such that the PET time activity curve will gradually reach a steady state. So where the slope of the curve is proportional to the cerebral metabolic rate of glucose. And now we have a task. In the case of a task-stimulated CMR glue changes, the time activity curve will reach a new steady state, manifesting as an altered slope. And then when task goes back, and the curve will return to the baseline level. So in this manner, functional changes in metabolism can be captured within a single imaging session. And we can also use a general linear model like what we did in fMRI to infer activations in fPET data. So just a quick comparison. So in fMRI, we know the baseline is roughly constant, and the signals will return to baseline after stimulus-evoked change. But the baseline of fPET signal is a roughly linear trend because FDG were trapped in the tissue, and the task-induced changes will reach a new intensity level following the stimulus. We're never going back. And we also need to notice timescale differences in the context of this study where we fMRI focus on seconds and fPET we're talking about over several minutes. So here's a demonstration of applying fPET-FDG to map visual activation from the early study, which is proof of concept. So compared to the prefrontal reference regions, you can see the time activity curve in the visual cortex demonstrates an activation. So we can identify a change in the slope when the flickering checkerboard is on. And the difference is more salient if we remove the linear baseline trend. So you can see every time the task, there's a slope change. So the validity of this technique has been demonstrated in several studies involving various sensory cognitive tasks, and also more recently in the resting state metabolic connectivity settings. So for the rest of the presentation, I will mainly just show the fPET FDG time activity curve post-detrending, just to help visualize the task or arousal effects. So having introduced the imaging modality, we conduct the experiments on a 3T Siemens scanner with a brain pad insert at the Martineau Center. So here's the picture and the experimental designs. And all the scans took place in the afternoon, and the subjects were partially sleep deprived to increase the pressure of them falling asleep in the scanner. And then to track dynamic changes in glucose metabolism, we reconstructed the PET data at a 30-second nominal temporal resolution. So for fMRI, we just use a very standard protocol. And then you can see it's kind of very big work size and TR considering nowadays the modern acquisitions, just limited by the hardware. So here it shows the summary of brain state changes during the scans across all subjects. So you can see most participants underwent stable wakefulness and sleep during the scans. As you can see, subjects tend to be more wakeful at the beginning of the session, and later as time goes by, there's increased likelihood of deeper sleep. So first, I will show that our trimodal measures can have sufficient sensitivity to measure arousal-induced changes in different physio processes. So for instance, here's a spectrogram of an occipital EEG electrode from a single subject. So we can use the power of alpha wave activity to infer subject's arousal state. As you can see, the subject shifted from wakefulness to sleep, and also have a brief wake during the session and then fall asleep again. So if we look at the fMRI time course, so consistent with previous reports, we can see a marked increase in fMRI signal amplitude in the sensory cortex during sleep. And for fPET dynamics, if we kind of remove the baseline trend based on the initial wakeful period, so you can see there is a reduction in the slope when the subjects fall asleep. And then you may see there's a brief increase when the subject becomes wakeful again, and then there's a decrease in the time activity curve when the subjects fall asleep. And in another subject, we can even identify altered metabolism across different stages of sleep. So these examples suggest that we can reliably detect sleep-induced changes in EEG, hemodynamic, and metabolic signals. So we then examine the first question. So whether these various physiological processes show coupled evolution over time. The one challenging we have here is that the hemodynamic fluctuation occur on the time scale of several seconds, whereas glucose dynamics unfold over minutes in our study. So to integrate information across different time scales, so we performed the following analysis. So because the amplitude of fMRI oscillations is a sensitive indicator of subject's arousal, so we use that as a metric of interest. So we tracked the dynamic changes in the amplitude levels by quantifying the time-windowed standard deviations of fMRI oscillations across a sequence of 30-second windows. And we chose this window length just to be consistent with the temporal resolution of the reconstructed PET signal. So here is an example from single subject's data. So the top panel shows how the amplitude of fMRI oscillations varies over time. And to mimic the cumulative nature of FDG trapped in tissue, so we integrate these fMRI activity measure over time to approximate fPET signals. And then we apply detrending to remove the baseline fluctuation. In this case, we just use a very simple third order polynomial detrending. So this will yield an fMRI modeled fPET dynamic signal. I was actually pretty surprised that this fMRI model signal closely predicts the actual fPET dynamics, can even capture very subtle changes in variation. So for instance, like highlighted by the red arrow here. So this I'm just showing a single subject's results. But if you summarize all the results from different subjects, so each panel here shows the results from a different subject, you can see the global fPET signal can be well approximated by these fMRI measures. So not surprisingly, at the group level, we can see a strong correlation between fPET and fMRI signals, with large hemodynamic oscillations emerging while metabolism declines. So the temporal coupling between hemodynamic and metabolic fluctuations may be attributed to their coupling with continuous changes in arousal. So my student Dakota helped me with this analysis. So we basically also quantified continuous arousal fluctuations using the ratio of EEG alpha to low frequency delta and theta power. So this is a kind of established way in EEG literature to track arousal dynamics. And we indeed observed a significant correlation between the EEG arousal metric with both fMRI amplitude fluctuations and the metabolic activity. So together, they suggest that multiple physiological processes are tightly coupled and well coordinated during the transition from wakefulness to NREM sleep. So how about the spatial dependence? So we next examined the spatial distributions of altered hemodynamic and metabolic dynamics. So to assess changes in fMRI oscillations, we estimated the amplitude of the fMRI signal for both wakeful and sleep states, and then we take the fractional changes of them and quantified which region shows the most pronounced increase in hemodynamic oscillations during sleep. And you can see this is a spatial map, and we can clearly see that in the sensory region, including the visual, auditory, and the motor cortices, the fMRI oscillation shows the largest during sleep. And for metabolic changes, we quantified the slope of fPET signal changes during wakefulness and sleep. If you recall, in a steady state, the slope of the fPET signal is roughly proportional to cerebral metabolic rate of glucose. And consistent with our large oscillatory activity, you can see the sensory network still show kind of only modest metabolic reductions during non-REM sleep. Whereas the higher order cognitive regions, particularly the default mode network, including the prefrontal regions and posterior cingulate cortex regions, they show very clear reduction in metabolism during sleep. So given the timescales of our measures, different timescales of our measures, and fMRI primarily captures fast oscillatory activity, whereas fPET-FDG in our application more robustly track those baseline activity over minute-long scale. So putting together, these measures kind of reveal a picture of two distinct patterns of network activity. So while most networks show reduced metabolism alongside increased fMRI oscillations, the sensory regions really show more prominent oscillations or alternations between high and low activity during sleep. And because of these large fluctuations, they also result in a relatively elevated metabolic baseline in these areas. And by contrast, the higher order regions are more suppressed. So what is the implication? We noticed that the emergence of large oscillatory activity alongside reduced baseline metabolism aligns with LC activity pattern reported in sleep in mice. So these burst-like LC activity have been considered to reflect periodical shifts between low and high arousability states. So a roughly 50-second cycle of alternations between heightened environmental awareness and offline internal processing has also been reported in several animal studies. And supporting the relevance of this to our data, we found those prominent fMRI oscillations in our data also peak at similar frequencies around 0.02 hertz. And consistent with animal literature, we also replicated the findings that the global fMRI signal strongly coupled with EEG sigma band activity and also cardiac oscillations, which have been linked to cyclic arousal patterns. So as for the spatial pattern, we also examine the spatial patterns of fMRI activity time locked to EEG features associated with sensory gating or autonomic arousal during NREM sleep. And we also found that the visual and motor area indeed show the most pronounced changes. When we look into the literature, these patterns, to some extent, have already been reported in previous studies. So taken together, these network-specific patterns might suggest mechanisms for sensory alerting or sensory awakening during light and intermediate stages of sleep, where periodic bursts of activity enhance sensitivity to external environment, allowing the brain to monitor for potential sensory cues that could trigger awakening. So a summary of the scientific findings here. So we first show that large hemodynamic oscillations emerge as metabolic activity declines during the descent into NREM sleep, and both of which tightly track EEG arousal dynamics. And these findings suggest that the physio processes do not evolve in isolation, but rather in a complex and well-coordinated manner across wakefulness and NREM sleep. And spatially, we identified network-specific functional and metabolic patterns during NREM sleep, particularly in the light and intermediate stages, where sensory networks show preserved metabolism and the periodic fluctuations between high and low activity, while high-order cognitive regions are suppressed. So this organization or pathway supports a potential mechanism for sensory awakening or alerting during NREM sleep. And finally, from a methodological standpoint, our data demonstrate potential for trimodal EEG-PET-MRI in elucidating neuro-hemo-metabolic states underlying a broad spectrum of cognitive and consciousness conditions. So this work, we have a preprint online, and it has just been accepted by Nature Communications. So hopefully it will be appearing online in the next few weeks. But for the rest of part of the talk, I want to talk about some further methodological considerations for this work where we put in the supplementary material, but it's really kind of important part for us to think through some questions to make the results more robust. So I will just go through each of them very briefly. So the first concern is that when we present the work, we always got asked whether because we wear EEG cap inside a PET scanner, whether we account for potential EEG cap attenuation. So when we start the experiments, we basically just start with what caps are available at our center. So we're using the EEG-fMRI compatible 256 very dense array geodesic nets. And the wires are copper wires, so it really causes attenuation. So we first did some like FDG hot phantom experiment to inject like FDG tracer into the fat water phantom and then compare the case of the PET signal intensity with and without EEG caps. We do see a slight reduction, about like 8%. It's not big, but there's a slight reduction in the PET signal intensity when the EEG cap is on. And we care about whether it will cause some structural noise in the PET images in humans. So we also perform a human experiment where we have the subject have a bolus injection of FDG outside the scanner. And then we take the subjects into the scanner. In one case without EEG cap, another with EEG cap, like spacing about like 20 minutes. And we account for the radioactive decays. So what we found is like we don't quite see very clear structure noise in the difference map between these two cases. And actually, because there are also subject arousal state differences, so the reduction we see in the phantom data are not clear in the human subject data. And if we segment the PET data into different brain RIs, so each dot here shows one brain RI, they roughly kind of, or to some extent align pretty well between the cases where subjects were an EEG cap and not wearing an EEG cap. And the more important consideration for our study is like we actually compare intra-scan contrast. So any attenuation effect will impact the sleep or wakeful states the same. Therefore, if we take the fractional changes, this pattern will be canceled out. So at the end, we didn't incorporate additional cap attenuation in our study, and we think it's not a major concern for our research. And the second one is, I mentioned several times, we remove baseline trend of the fPET time activity curves. In the sleep study, we use a very simple way, just use the third order polynomial detrending, but this may have a little bit caveat. So this is relates to a study done by my student, Sean Kersey. So I will first provide some background. So we know like nowadays with fPET, we can analyze the task evoked effects in a manner similar to fMRI using general linear model, but there is some more issues for fPET here. So for fMRI, we know like the baseline is in a very like low frequency trend where the task regressor or task effects are on several second scale. So they're really in different frequency. So even if you don't model the baseline well, you wouldn't see much influence on your task activation map. But for fPET, it's not a strict linear. And then sometimes when you have like bolus injection, it has a pretty like dynamic phase at the beginning of the scan. But then your task effects also on a several minute scale. So in this case, there might be concerns of covariation of the baseline and then the task effects. So how to properly address the baseline is more issue of dynamic PET studies. So in existing fPET FDG studies, sometimes people are using lower order polynomial detrending, and sometimes people are using global signal either by fitting low order polynomials or restricted to prior knowledge of tasks in active regions. So yeah, my student Sean, he first started going ahead to see whether there are consistent patterning kind of due to imperfect baseline removal. So we use a very nice resting state of PET FDG data from Monash University, and then each gray line here shows the results of a single subject, and then here shows the mean time activity curve. If shows the residual time activity curve if you do mean tag regression or use low order polynomial. So you can really see there's some like residual patterns that are consistent and systemic in the time activity curves. And if you contrast them with simulations, you can see it aligns with the measured data very well, suggesting that this pattern really due to the intrinsic tracer kinetics. So the challenge here is like if you have a stimulus on a very low frequency scale, and if the stimulus is correlated with the residual pattern to some degree, even it's very tiny, but if you have a lot of subjects, it's going to show a systemic significant group effect. So we just tested, this is a little bit inspired by Eklund paper in the fMRI field. So we basically apply sham test regressors to the resting state fPET data. In a perfect world, we shouldn't see any activation because the subjects are in a resting state. And then we first used the third order polynomial detrending as an example. So you can see here are 3 different types of regressors. And the bottom shows the group level activation map by applying the sham task regressors in a general linear model. And it's a bit striking to me with a number of like 26 subjects, you can already see robust or significant activations with some regressors that strongly correlated with the baseline. And even for global regression, while the global mean is 0, but then there are regional variability in the residual patterns. So you can see using different regressors, you can still see significant activations or deactivations just due to the baseline imperfect removal. And then while globally, they may sum up to almost 0. So this is a concern that actually makes me think about our sleep-wake study, whether some of them may be attributed to imperfect baseline removal. So we did some control analysis. So we have some subjects didn't fall asleep during the scanner, so they can be used as a null condition where there's no prominent arousal effects. So what we did is we then apply the sleep-wake timing or the true sleep-wake timing inferred from all subjects to these null data. So we have like around 20 subjects. So we apply all these 20 subjects sleep-wake timing to these null data sets. So we get a new batch of 20 subjects data, and then we look at the group level activation. But we do see quite distinct patterns compared to what we see in the sleep effects. And we also tried a few different subjects, and the results still not consistent as what we see in the real data. And the reason is that on one hand, sleep is just such a strong effect, might dominate than the systemic bias. But probably more importantly is just because each subject have different sleep-wake timing. So it's kind of jitters somehow will cancel out those systemic effects. So this is just a second question we're considering. So that's why we trust our results are not really impacted much by the baseline feeding. And a third one consideration we had is like the accuracy of CMR glue quantification. If you recall how we did that, we have a very simple approach. We basically just assume within each sleep and wake segment, the subjects will fall asleep and they will reach a steady state where the FDG tracer in the plasma almost become constant. And then in those cases, the slope will be roughly proportional to the cerebral metabolic rate of glucose. And then we take the ratio between those. And to restrict the data to more stable wakefulness and non-REM sleep, we only take the segments longer than 5 minutes. But still, this is not so long enough for them to reach steady state. And in fact, later when we have different studies, we found like it's really never kind of the plasma will reach a constant state. So like Sean also helped me try a padlock approach where it would be less sensitive to this such a strong assumption. And then in this plot, like we can definitely see in both segments, it later become more kind of linear. But still, if the scan time is just like close to 5 minutes or longer, or shorter, and then the feeding wouldn't be perfect. But in any case, if we use a padlock plot, we can still see very strong similarity of the derived metabolic patterns compared to what we achieved with a simple slope-based ratio calculation. But then we also notice like the percent signal change estimated here is a little bit stronger than what we report in the main text. So this is basically suggesting these oversimplified ways of calculating the fractional changes combining with not so long stable awake and sleep states, making the exact quantification value may not be that accurate, although the qualitative changes are reporting the sensory region have more preserved metabolism and the higher order regions have more suppressed activity still holds. And the final consideration we have here is about network-specific activity in deeper non-REM sleep. So we already talked about how, and emphasized a lot about light and intermediate sleep where the sensory region remain active and to enable sensory awakening or alerting. But we know when people enter the deeper non-REM sleep, it's hard to be waken up. So what might be the mechanisms? So unfortunately for our data, we didn't have too many subjects like transition from stable light and intermediate sleep to deeper like N3 non-REM sleep. But for those subjects, it can already provide some hypotheses that can be tested in future studies. So for instance, this is one subject where you can see from wakefulness and then to intermediate light sleep and later to stable N3 sleep. And if you look at our fMRI time course in both the visual network and the global signal, you can actually see when people enter deeper sleep, the magnitude of these fMRI fluctuations start to decrease again. And then if you look at the fPET time activity curves, it will continue to reduce. So this basically suggests the possibility that both hemodynamics and the metabolism continue to go down in the non-REM sleep. So taken together, the current picture we have is like there's dynamic changes in the non-REM phase, where in the light and intermediate stage, the sensory region remain active. They show periodic high and low activity to facilitate alternations between heightened environmental awareness and offline processing. But in deeper sleep, it's like all this network goes down, the activity. But this is just a hypothesis pattern which remain to be tested in future studies. Okay, so a summary of these further technical considerations. I first show EEG-CAP used in our study could indeed cause modest reduction in the PET function counts, but we don't see clearly visible structural noise in the data. And since we only focus on the intra-scan comparisons, the fractional changes between sleep and wakefulness, so we think it's a lesser concern for our study. Second is like regarding the potential artifacts introduced by baseline specification, because sleep is such a strong effect, and also the sleep-wake timing are jittered across subjects, so we don't see a strong systemic effects manifesting at the group level. And the third one, regarding the accuracy of CMR glue quantification, yes, because we have a bit, we try to use the data from as many subjects as we can because it's really hard to acquire the trimodal data, but then it still have issues of really not ensuring the tracer enters steady state. So ideally, combining parallel plot with a longer sleep-wake segments may get more accurate quantification, but the qualitative changes still kind of hold. And finally, our results do suggest some interesting and altered patterns of network-specific activity in deeper non-REM sleep. So it'll be interesting to pursue this further with longer scan time. Okay, great. Yeah, thank you. So if you have any questions about the talk, feel free to reach out, and I'm also happy to take any questions you might have.
Yes, thank you very much, Dr. Chen. It was really interesting talk.
Yeah, thanks.
Until we wait for questions in the chat, I would like to make a question. So you comment, you reconstruct the data from PET, fPET in 30 seconds frames, right?
Yes.
So did you reconstruct this 30 seconds for the whole time course?
Yes. Oh, do you mean like for the entire 90-minute scan, like every 30 seconds, we have a data point?
Okay, and then, yeah, and then
Yes.
when you evaluate these lobes, you consider just more than 5 minutes, right? For the
yes. So we only consider stable wake or sleep, stable sleep that's kind of longer than 5 minutes.
But then you exclude the other time points, or okay?
Yes, we'll exclude those time points.
We have a question in the chat, so thank you very much for your wonderful sharing. I have a curious point, although this may deviate somewhat from the core findings of this research. I'm curious about whether blood glucose and mitochondrial phenotypes such as density or energy transformation capacity both reflect metabolic activity, especially energy consumption. Can I expect mitochondrial changes to have similar spatiotemporal variations patterns as the blood glucose changes you observed, or do mitochondria and blood glucose reflect different aspects of energy supply? I wonder what insights you might have on this question.
Yeah, thank you for the question. Thank you for the question. At first, it's like I really don't know too much about the mitochondrial function well, so probably my answers wouldn't be really helpful. But my understanding is like glucose is just one aspect of metabolism, but mitochondria, I think, is like upstream metabolism and everything also related to that. And there have already been evidence in animal literature that the sleep-induced changes in oxygen metabolism and glucose metabolism are not proportional during sleep. And then this could be, if I recall correctly, like the brain will be more oxidative, the metabolism become more oxidative during sleep. So I would imagine at least like the quantitative changes will be different, but I don't know about the spatial temporal structure. It'd be interesting to pursue this question. Sorry, I don't have further insights, but this is pretty good point.
So I cannot see any further questions.
I think we can just ask question directly if you have any questions.
Yeah,
So
sure.
sorry, I just I want to ask. So thank you very much for the talk. I think so the first part of the analysis of temporal coupling, you are doing all the ROI using visual, right? And you are
Yes.
saying about this arousal. So can you how to define this arousal? You seem to suggesting that it's kind of arousal to the environmental stimulus. But is that, for example, usually you're talking about some subcortical regions, like like thalamus and some other region may also contribute to arousal. Have you examined those regions, or do you see any like findings in the subcortical nucleus, for example?
Yeah, I think that's a pretty good point because ideally, like the sensory gating will be through the thalamus, kind of controlling for that. So, we currently just taking that as a whole because it's limited by the spatial resolutions what we have. But then, especially for PET, while we reconstructed at like 2.5 millimeter, but then the point square function, everything making it be probably really hard to look at different structures within the thalamus. So, we have a separate study kind of now doing, we just trying to really look at like spindle and K complex and how in the thalamus and also including cerebellum, all this region will be different. So it's just kind of ongoing, but I do admit with this data, we're limited by the resolution. It's hard for us to see very detailed patterns on the heterogeneous activity within the thalamus.
May I also ask another question?
Yeah.
I imagine you had some motion during the study because between sleepy and not sleeping phases. So how did you handle motion for so short 30-second frames?
Yeah, I think that's a pretty good point. So we did a couple of things to mitigate emotion. So first, the subjects we recruit, they have like prior PET-MRI experiences before. So it's kind of, we have the data, get an idea how much motion there will be also there. We're familiar with this environment. A second is like we do motion correction. It's because FDG really have whole brain coverage. We just do volume-based registration after the data acquisition. That helps a bit. And also finally, it's because like we are looking at the metabolic changes along quite long scale, and then the metabolic declines, a pattern will be quite different than the oscillatory or whatever caused by motion. So in fact, when we found like the motion impacts on the fPET are not as that prominent in cases like fMRI. We do the correlation or everything. Yeah, so that's kind of how the approaches we've taken to mitigate head movements. But we do later have like more aging population. We found it's issue harder to address. So we are working on MRI guided motion correction because the benefits of doing them simultaneously, we can estimate the motion very precisely from simultaneous MRI and incorporate it into PET reconstruction.
Okay, thank you.
Mm-hmm.
So we have more questions now. You mentioned in future research you want to investigate deep sleep. How do you foresee inducing a higher rate of deep sleep in participants?
Yeah, I think that's a good point. It's a challenge, right? Because it's limited by how long you scan, subjects can lie in the scanner, and also by like the radioactive decay time, half-life time of the tracer. So it's a bit hard in the PET-MRI environment because MRI is so noisy. But then we have our center is going to have a whole body PET CT, and I was thinking in these high sensitivity scanners, we could increase the scan time, and also because it's more quiet, so it will make possibility to increase the likelihood of including deeper sleep.
We have another question. Thank you for the very insightful presentation. Congratulations for this amazing work. Is the study group homogeneous in terms of age and gender?
Yeah, so I think two questions. Age, this is mainly like from 18 to 36, maybe a few subjects slightly older. So in general, it's like a young, healthy population. So we didn't particularly look at the age effects, but we do have an active study where we are acquiring aging and we encounter a lot of challenges of having both sleep and also motion issues. Yeah, but that's definitely of interest. And for gender, for this data sets, we have two. I think not. Yeah, we do have more like dominant female subjects in the data, but we also report the results of both female and male. Their spatial pattern look pretty similar, and I think in the supplementary material we have it, but we didn't do a deeper investigation about the particular gender effects. Yeah, I think that may kind of require a larger population to do that. Yeah, I also mentioned why we have a bit more female. It's also because we limited by the hardware. We need to wear EEG cap inside scanner, so somehow limit the head size. So that kind of is one constraint.
If I may also, can I ask another question?
Mm.
Yeah. So so your results show that fundamentally the dissociation of the sensory and then the deformal network, in one is in PET and another one in fMRI.
Yeah.
I'm a little bit. Can you explain a little bit about your model? You show that kind of pattern, why that will happen. You treat the PET as a baseline, and then the
Yeah,
oscillation. And also, I'm a little wondering.
This one?
Yeah, something like this. And I'm just wondering, so you show the very high coupling between this amplitude, fMRI amplitude, and the PET, the metabolic activity. So
Yeah.
that is only happens but based on your model, that means that the result you showed is in the visual cortex, right? But if you examine the deformal network, whether this relationship are still hold, or can you explain this slide a little bit?
Yeah. So I think the global, because there are two cases. There is one global effects. There are also like region-specific effects. But then for both of them, while I keep highlighting like the sensory region goes between high and low activity, but then actually in higher cognitive region, we also see this oscillatory activity of high and low. It just extent is much smaller. So in fact, like in Solomon's like as a whole and cerebellum, we also see these patterns. So it's kind of globally we will see during sleep there is emergence of large oscillatory activity and also reduced baseline activity. It's just a relative extent will be differ between different networks. So the plot I'm showing here is really global. It's global, yeah, global patterns. And then yeah, I don't have the figure here, but we're also using the global fMRI regress against the regional PET signals, and you can see a lot of regions just like show strong coupling with that. So in general, the pattern of coupled evolution of emergence of large hemodynamic fluctuations and reduction in metabolism, it's a kind of, you can see a widespread across the brain. Yeah, and for here, more like it's somehow they are coupled because when you have this large oscillatory activity, because the high close to the wakeful cases and low is born here, so that's why the baseline seems higher than what you in higher cognitive regions, because the oscillatory activity maybe their high level or didn't really reaching like the wakeful periods. So as you can see, if we use more robust MR methods, let's say maybe multi-echo, those kind of can better handle those drifts, and maybe you can separate those baseline activity and oscillatory activity. Maybe you would be able to see similar phenomenon. That's just my guess. Yeah, but there's by doing that, I'm assuming there's no fundamental changes between oh yeah, that's oversimplified. I just commented the oxygen metabolism and glucose metabolism might be slightly different. Yeah. So that will go to more deeper into that types of questions.
Okay, thank you.
We have more questions. Thanks for your interesting talk. Have you ever thought about including patients with sleep disorders and compare their both PET patterns?
Yeah, that's a pretty good question. We haven't, but because we have quite rich kind of data here. Yeah, especially one direction what we were thinking is like for fMRI, we're only just talking about like the hemodynamic oscillations, but then there is emerging interest, a lot of talking about like using fMRI, you can have this focusing on the inflow effects in the fourth ventricle, you can see CSF activity that related to lymphatic clearance. And then in our manuscript, we also correlated that with metabolism because we're thinking maybe metabolism more related to waste production. So then have this coupled activity. We do have some assumptions how that will change in neurodegenerative disorders. But we haven't really acquired those data. So far, we're just focusing on aging and people have genetic risk for Alzheimer's disease, but it's just ongoing study. We haven't extended to other sleep disorders.
And the other question is, great work. Could you use another EEG cap with less electrodes, like 64 electrodes, or this high coverage of 256 electrodes is needed in such connectivity studies?
Yeah, that's a good point. As I mentioned, we started with 256 is basically what's available at that time, kind of at our center with MR compatible caps. But ideally, you have you probably just need like 6 electrodes or so to infer subjects' arousal states. So I think if those caps are available, you can certainly use them. One considerations when we have also, while we have 256 electrodes, a substantial amount of them, we were basically used as sham electrodes, just as a reference layer approach to remove EEG artifacts, like MR artifacts in the EEG signals. So we do, for the particular processing perspective, we want more electrodes. Yeah, but ideally, if you don't think about denoising or any other issues, you can certainly with fewer electrodes.
So I cannot see any further question at this point.
Yeah, thank you all for the question.
Yeah, thank you. It was a really nice discussion.
Yes, great. If there are no other questions, can I ask more a little bit of? So, do you think that what temporal resolution can we get like for higher temporal resolution. So you are now using 30 seconds. Is that possible you're doing like shorter and shorter? And what is the temporal limit for that? Do you have any thoughts?
Yeah, I think that's a pretty good point. I guess there are two aspects. One is like the intrinsic. I would imagine that biologically the metabolic process can be super fast. Like I'm not talking about that, but one is like whether how fast can fPET FDG track, and another aspect is like how fast can we get a high sensitivity measures of like very high sensitivity reconstruction. I think that's easy to answer. That probably depends on the hardware and the sensitivities, the sensitivity of the scanner. So you can certainly push. And I know a lot of groups like Andrea's Hands group and like Shanna's group, they push into second scale reconstruction. So that's totally feasible. But for the mechanism, so very rapid fPET FDG oscillations, I kind of feel it's still potentially in debate, because for us, we can see very robust metabolic changes on several minute scale, and earlier when introduced at the Martino Center, people also did some tests using gas challenge to see whether their metabolism changed, because there are concerns whether some of them are due to blood flow. But so similar experiments hasn't been validated for shorter time scales, but there have some ideas, I think, from a recent opinion article by Andreas Chen was suggesting that super rapid oscillations may not related to phosphorylation of FDG, but it's more related to metabolic supply. So I kind of feel that's still a kind of under debate area. People definitely report very fast FDG dynamics coupled to stimulus in the sub-minute scale, but then whether it's related to glucose consumption and which facets of glucose consumption, I feel it's still an open question. It's just my personal opinion. Yeah.
Yes, thank you. But then, so have you considered for them using other tracers that for them using carbon 11 so that it's faster? So, I mean, measure different types like neurotransmitters. I think we have a review paper, like a lot of study on that. So.
Yeah, that's a pretty interesting direction. Certainly, I think it would be fun to do neurotransmitter studies. It's just like, we haven't, like my lab haven't started doing that. I think but there are other labs at the Martino Center doing it. I think the challenge is like both a half-life time of them and also how strong effect size, because fPET FDG just somehow very robustly we can see this altered metabolism, and for functional changes in the neurotransmitter system, like it has always been challenging. But we are hoping with the new sensitivity scanner, and we will be able to kind of go into that direction, because that would be really exciting. Yeah.
Thank you.
So I cannot see any further questions from the audience.
Yeah, just feel free to ask questions if you have anything to discuss. Really, thank you for the very wonderful talk. I feel it's very, yes. So you think that those subjects, so within like 1 hour or so, they can fall asleep, right? That's kind of very surprising to me a little bit. And
oh yeah, actually not too surprising. I think there are some studies, even just resting state fMRI. I think in the group level, they show people around like 20 minutes. A lot of people, I think very high probability of people falling asleep. I think the kids even just a bit drowsy, it's already in stage N1 sleep. Yeah, and on our subjects, we also have a sleep deprivation. So they were sleep deprived 4 hours to 4 hours the night before. That can cause that. Actually, I need to acknowledge it might be a slight limitation here, is because people are sleep deprived, their baseline cerebral metabolic rate of glucose is a bit lower in that condition during wakeful periods. Yeah.
Since you comment on this sleep deprivation, so how you monitor them during this part?
Oh,
If
you mean the night before?
Yeah.
Yeah, no, we didn't monitor them. We basically just have a questionnaire so they fill out how many hours they sleep. So in the case we do have subjects end up like staying awake for nights. Yeah, we have the questionnaire like reported in the paper. You can see it's really hard for people to control like how many hours they sleep. But in our case, it's just like in general, we just treat them all together, have a wakeful period and then the sleep period. Yeah.
Okay, thank you.
Yeah, and also a trick I learned was like you tell subjects, don't tell them this is a sleep study. It's easy to fall asleep. You just say like it's okay, like it's this is a study where you're lying quietly inside scanner. It's okay, stay awake all the time. It's also okay you fall asleep. So they wouldn't feel stressful.
Really?
Yeah.
Then it means that all subjects are bad subjects. You do not tell them to sleep, but they fell asleep anyway.
Right. Because a 90-minute scan, it's really hard not to fall
It's
asleep.
hard to control.
Yeah, we also have cases like initially we do an MPRH like anatomical scan before doing the FDG tracer, and then people already while we start have EEG acquisition, you can see they already fall asleep. And for some subjects, we do have a lot of not a lot, a few subjects where don't have stable wakeful period and have all drifting between like N1 and deeper sleep. So yeah, we're still struggling to find the sweet spots. But in general, I feel, yeah, that's why I kind of feel if we don't care too much about MR portion, just in a PET CT scanner, it would be easier, but it would be more quiet. And we don't deal with also potentially cleaner EEG signals.
Okay. Thank you very much. We are about time. Thank you for the talk, Jingyuan, and thank you for the moderation. And thank you for all who comes. So it's about time today.
Okay.
See you next month for a serious talk.
Okay, thank you.
Okay, bye-bye.
Thank you all.
Bye-bye.
Bye.